Hi guys, my name is Q, and in this lecture I'll be going through meiosis. So before going to meiosis, I have to define a few terms. First is haploid and second is diploid. So I'll first talk about diploid. So diploid cells are cells with 2n number of chromosomes. In other words, there's going to be a pair of each chromosome. So chromosome number 1 is going to have 2, which is one pair, and that's going to be the same for all the other chromosomes. For the case of haploid cell, it's going to have n number of chromosomes. In other words, chromosome number 1, there will be only one. Chromosome number 2, it will have only one. And yes, the function of diploid cells are same as mitosis. So it's usually going to be for repair of the damaged body parts, growth of the organism. And for haploid cells, it's literally just for reproduction. These kind of cells, the haploid cells, are called gametes, and diploid cells are usually called as somatic cells. No, not usually, they are called as. Oh my goodness. Bleh. Yes. Okay, so when two haploid cells, okay, let's see, haploid cell number 1 and haploid cell number 2. So for example, this is the sperm cell and this is human egg. Okay, so when these two fuse together, we're going to have a diploid cell, which is called zygote. And note that zygote refers to a fertilized cell with which is only a single cell at the moment. So when it becomes 2 and onwards, technically it is kind of zygote, but not exactly a zygote, should I say? Yeah. Okay, so now I'm going to compare the differences between mitosis and meiosis. Okay, so the first thing is the resulting cell. So in the case of mitosis, it's going to be a somatic cell. In the case of meiosis, it's going to be a gamete cell. Recall what's the division of somatic and gamete cells in the start of the video? Okay. The next one is number of divisions that's going to occur. In the case of mitosis, there's only going to be one, but in meiosis, that it is going to occur twice. Okay, in the case of whether there is pairing of homologous chromosome. Okay, so pairing of homologous chromosomes. So what exactly is this? So this is a pair of homologous chromosome. So these two aligning at the metaphase plate is called pairing of homologous chromosome. In the case of mitosis, no, this will not occur. In meiosis, yes, this does occur. Okay, number of daughter cells. So if we have an initial parent cell, in the end, how many cells are going to have? In the case of mitosis, we're going to have 2 because it's duplicating. In meiosis, we are going to have 4, okay, because it's twice the division. Yes. Final number of chromosomes. So in the case of mitosis, so it is just duplication. It's going to be the same, which is 2n. Meiosis is going to be halved. That's why it's going to be n. And the genetic material compared to the parental cell. So in the case of mitosis, the genetic material will be same. The daughter cells are going to have the same genetic material as the parent cell. But in the case of meiosis, they may be different. Well, usually they are, but technically they may be different. Okay. Okay. Lastly, uses of mitosis and meiosis. Yes. This is actually mentioned earlier, but if I just repeat, mitosis, it is used for growth development of the organism and repair of damaged parts. Meiosis, it is going to be reproduction. Okay, so now I'll go into the process of meiosis. Okay, so in the case of meiosis, you can actually think of it as mitosis just times 2. So it's like 2 consecutive mitosis in one procedure. But there is a key difference, and this is one of the most important differences that you have to take note. Okay, so we're going to take a look at mitosis metaphase and metaphase of meiosis 1 and meiosis 2. Yes, of course, meiosis is separated into meiosis 1 and meiosis 2, which consists of each consisting of prophase 1, metaphase 1, so on and so forth, and then again prophase 2, metaphase 2, so on and so forth. Okay, So in the case of metaphase of mitosis, it is the sister chromatids that is going to align at the metaphase plate. So in other words, it's going to look something like this. Okay, so at the metaphase plate is the sister chromatids, sister chromatid. Yeah, so these are going to align at the metaphase plate. But in the case of meiosis 1, metaphase 1 of meiosis 1, it's not going to be the sister chromatids, it's going to be the homologous chromosomes. In other words, if I'm going to have a metaphase plate here, we're going to have one chromosome here and another chromosome here. Oh, just for information, these two are of the same chromosome number, so it's going to be chromosome 1 and chromosome 1, 2. The 2 is TOO, by the way, just in case. Yeah. Okay, in the case of meiosis 2, meiosis 2, the metaphase 2, this time it's going to be again sister chromatids. So again at the metaphase plate, it's going to be the sister chromatids that's going to align back there. Okay, so now I'm going to take a look at anaphase of meiosis. Yes, for during an anaphase of mitosis, it's going to be the sister chromatids again. Sister chromatids that are going to move to the opposite poles of the cell. Remember, poles, for no ends, poles. Yes. And in the case of anaphase 1, it is going to be homologous chromosomes again. Okay, so it's like oh my goodness, my terrible drawing skills. And in anaphase 2, it is again going to be sister chromatids. Okay, and so what is the difference between the anaphase of mitosis and anaphase 2 of anaphase 2 is that the number of chromosomes that is going to be different. Okay, so this is the difference between the metaphase and anaphase of mitosis and meiosis. Okay, so initially, if we have 4 chromosomes, too long and too short. Okay, so in mitosis, it is going to be something like they're going to split, and in the end, we're going to have something like this. In other words, the last cell is going to have, the daughter cells are going to have like this. So these are 2 cells we're going to have. In the case of meiosis, recall that in meiosis 1 we have homologous chromosomes that aligns. This is going to the whole thing is going to split. So in other words, after meiosis 1, the number of chromosomes will decrease, and they're still at the kind of like the replicated state. Okay. So what happens after this is the same as the mitosis. These sister chromatids are going to separate on long and short, on long and short, and the same thing will apply for the other cell as well. So the key difference is that in this case, the number of chromosomes will still remain the same, but in the case of meiosis, the number of chromosomes will actually be halved. And technically, the amount of DNA molecules in the entire cell has halved, but the number will still remain the same. After meiosis 2, the number of chromosomes will remain the same, but the amount has halved. So this is kind of the tricky part, but it shouldn't be that difficult. Okay, so here's the second major part of the meiosis. Okay, the thing is genetic variation of through the meiosis. Okay, so like genetic variation means that when it passes from one generation to another, like from the parental to F1 generation, how does it achieve difference in genetic information they have? Okay, so the first thing is going to be independent assortment. Okay, so Okay, so this happens at metaphase 1 of meiosis 1. Okay, so what it happens is that initially, okay, I'll use, okay, I'll use alphabets, A, and B. Okay, so this is the one that we have the parent, and uh, depending on how they align in metaphase 1, after meiosis 1, which one gets separated is different. So in the case 1, we can have big A, big B, small a, and small b. So in other words, these are going to become big A, big B, big A, big B, and so on and so forth for this one. So this is one kind of combination we can have. Okay, in the other case, we can have a line. Okay, actually, I did it wrong. We can have the alignment of big A, small a, and then small b, then big B. So when this separates, it becomes big A, small b, and small a, big B, and so on and so forth. So this is the kind of difference. This is the kind of difference that we're going to have in the different gametes we're going to have. Okay, and the second one is crossover and crossover and recombination. Okay, so what exactly is crossover? So this occurs at prophase 1 of meiosis 1. So what happens is that the two chromosomes of the same number, for example, chromosome 1 and chromosome 2, are going to align next to each other. Remember, this is not metaphase yet. It's like before they're on their way to like aligning at the metaphase plate. So they'll move around, and there will be a case of the spot where they have the same loci. So it's like, for example, if we're going to name the top part as base pair 1. So for example, this part is going to be base pair, let's say 200. Okay. So in other words, it's going to be it has to be the same spot for both chromosomes, and it will occur only between non-sister chromatids. Remember, these two are sister chromatids, but this one and this one are not sister chromatids. Okay, so the point that they're actually touching is called chiasma. Chiasmata. Sorry, chiasma was something else. Yes. Yes. So if I just summarize the whole thing, this chiasmata is only going to form only when it has the same locus, which is the same spot within the DNA slash chromosome. And so what happened is that, okay, so let's think of something. Okay, for example, in one chromatid, so this is one chromatid we have, and this is the other chromatid we have. Okay. So what happens is, okay, I will just take a look at this spot. Okay. So from like this, so these two parts are going to break. Well, if they're touching, but it's like, so it swaps apart and becomes like that. So this process is called crossing over, and when they combine back to the original, not original, like the main piece, this is called recombination. Just for information, they can also be crossing over happening at here and here as well, as long as they're in the same loci. So what's going to happen is that it might end up like this. So technically speaking, crossing over can occur anywhere and anytime, and the number of crossing over that can occur is totally random. In other words, we don't know whether it will occur or not. Okay, according to our professor, And in our syllabus, we assume there's around one crossing over and recombination in the process of the entire meiosis. Yes, that's the estimate we have in our syllabus at the moment, yes. But in reality, like what I said, we have no idea. I will swear just now. Okay, so the third part is random fertilization. So this is actually, I think it's one of the like most unpredictable and like the simplest and yet the most effective in a way, so in a sense. So yeah. So in other words, recall that through independent assortment, we can have like different combinations. Okay, I should do this. Different combinations. Okay, so from the mother, we can have different like out of the um, four possibilities we have. So we can have one of them, and from the father, we can have another kind of possibility, like big C and small d, for example. So in other words, and out of these four, and one out of four and one out of four, and considering that crossing over could occur as well, typically there's like uh, in human body, they say there's around few 100 trillion ways that like different ways of these kind of combinations. In other words, random fertilization is just random fertilization of um, different possibilities of sperm and different possibilities of egg. And when these like fertilize into a zygote, we have no idea what the zygote is going to be. Isn't that pretty awesome? Alright, so that sums up for the meiosis, and thank you very much.